want you to consider my pen as a genome. Your whole genome, okay, your body's or any organism's whole genome. Just consider it like that. We'll study the different categories of genomes using a pen. Okay, so structural genomics involves the physical nature of genomes and includes the sequencing and mapping of genomes. It deals with the study of the structure of entire genome of a living organism. In other words, it deals with the study of the genetic structure of each chromosome of the genome. It determines the size of the genome of a species in megabases. MB, wherever I am using MB, it is megabases. Wherever KB comes, it is kilobases. Okay. And also the genes present in the entire genome of a species. Okay. So now what is with this pen? Well, consider this pen as an entire genome. What is the physical structure of it? Physical means whatever you can see outside, whatever is expressed outside. Okay. Now this pen, if you look, it is a long structure, rigid structure. Okay. It is made up of certain plastic and certain metal parts. Okay. Inside it you have a refill filled. So these are all what? These are all the physical nature of this pen. Same way, while we study what is the structure of chromosome, where is it located? For example, in this pen, this button is located towards the end to push it, right? Likewise, where are your genes located? Inside your whole genome. Your DNA is too long, right? Where is that gene located? What is the structure of that gene? Okay, all those studies, the mapping is like you use Google Maps. Where is what? Correct? Same way we have mapping of genomes. Where is this genome located? Where is this gene located on your whole DNA? Okay, where is this junk piece of DNA located? All that is physical. So the structure or the physical nature of genome, that is what we'll study in structural genomics. Okay, and we'll be also looking at the size. For example, this is this much long, 10 centimeter long pen. Likewise, what is the size of the genome? We're not only talking about human genomes, we are talking about all the species, whichever we want to study. Okay, so that is structural genomics. Next is functional genomics. Functional genomics, this is the second category we are talking about. Functional genomics studies the functioning of genes and metabolic pathways. That is, the gene expression patterns in organism. It deals with transcriptome and proteome. Now, what is transcriptome? It refers to the complete set of RNAs transcribed from genome. That is, you might have read about the transcription process, right? From DNA, we get RNA by the process of transcription. So from that, you can understand the term transcriptome. Transcriptome refers to the whole set of RNAs. Okay, so basically we're giving terms to the whole set. Genomics is a wholesome study, right? So like that, transcriptome is the whole set of RNA. Okay, so in functional genomics, we will be seeing that we'll be dealing with transcriptome and proteome. Proteome is what, as the name suggests, it refers to the complete set of proteins encoded by a genome. Okay, so again, coming back to my pen, how can I use my pen to tell you what is functional genomics? Consider this as your whole genomics. Now, what is the function of this pen? Okay, function of this pen in simple terms is to write something. You want to write something, you can use it to write. There are different things you can do with a pen. You can play pen fight and things like that, but I'm talking about the basic function. Okay, so you can write with it. So that is the function. There's ink filled inside this with which we can write. So all those little details that we are using, you know, that what it does. This pen is there, that is your structure. We know the structure. What it does is the function. Same way, what our genome does, it makes RNA. From the RNA, you have protein. So what do those proteins do in our body? So proteomics is kind of a part of genomics only, functional genomics, because we study about what proteins do, okay, in this also. So they are both linked together, functional genomics and proteomics. We call them together, okay. So I hope you understood what is functional genomics. The function, or the functioning of your genes, the pathways, all those gene expression patterns, everything comes under functional genomics. Next is comparative genomics. Now, what is comparative genomics? Comparative genomics involves comparing the genomes of different organisms. From the name itself, it's very clear what is functional genomics, right? I'm sorry, comparative genomics. Comparative genomics can be used to define important structural sequences that are identical in many genomes and to detect evolutionary changes across genomes. Studies of comparative genomics have already proceeded far enough to reveal that in related species there is considerable synteny, that is conserved gene location within large blocks of genome. So now that's a huge text over there. How can I explain it with my pen? Okay. For comparative genomics, of course, what you need is another pen because you're going to compare one genome with another. You have human genome, you have chimpanzee genome, for example. Okay, so considering these two pens, what is the difference between these two? What is the similarity between these two? Both things you'll be studying. You see a word over here, synteny. Synteny is nothing but the conserved gene location with large blocks of genome. That means in both of this, something is there which is not changed. Okay, though these both look different, but something is conserved in this. That is called synteny. For example, you can say that this uses plastic and metal for its body. This also has a metal and a plastic body. So that is synteny. That is like the conserved part. Okay, so when you're saying what are the differences in this, totally the difference in shape, the difference in how it writes, okay, the functions as well as the structures, okay, we'll be seeing that. So that is what is comparative genomics. I hope it is very, very clear right now, okay. If some of you are feeling it is like I'm teaching small children, don't feel like that, because I feel that, you know, when you learn a concept, get as deep as you can and make the basics clear. Then you don't have to mug up these. Whenever somebody asks you what are the broad categories of genomics, remember this pen, and then just go ahead, okay. Tell them the 4 categories. So the next category, what is it? Mutational genomics. Mutation, again with the name you'll be able to understand. Mutational genomics is the field of genomics that characterizes mutation associated with the genes, okay. It studies the genome in terms of mutations that occur in a person's DNA or genome. What are the mutations occurring in a person's DNA? That study, okay. Mutational genomics links genotype to transcriptional state. In mutational genomics, the difficult task is to identify the genes causing transcriptional changes. Okay, so that is the text part of this, or the explanation part of this. Again, coming back to my pen. How can I explain this to you with my pen? Consider this is my genome. Okay. After some time, I notice due to some mutation, it becomes like this. There is something coming and joining over here. It has mutated, right? Now, mutational genomics is study of that. In which gene of this genome the change occurred that something like a cap was added to it. This change has come over. So that is what we'll be studying in mutational genomics. Now, in order to understand the structure of a genome, okay, we need to do some analysis, right? For example, you have your pen, you need to find out a structure, you need to do some data on it, right? Data finding on it. That is called genomic analysis. So that is what we are going to start. So what is genomic analysis? Genomic analysis is the identification, measurement, or comparison of genomic features such as DNA sequence, structural variation, gene expression, or regulatory and functional element annotation at a genomic scale. Methods for genomic analysis typically require high throughput sequencing or microarray hybridization and bioinformatics. Now this high throughput sequencing and microarray, all this will come in the second module or second unit of your this thing. Basically it is high throughput means to get higher efficiency and higher volume of genome is considered. Okay, that is high throughput. Now, some of the questions which biologists want to answer today using genomic analysis, or why do they need genomic analysis? First of all, given a DNA sequence, you have a DNA sequence with you. What part of it codes for a protein and what part of it is junk DNA? So you have a whole genome. You should know that our DNA, it has some genes and others are DNA sequences which do not code for a protein. That is called as junk. So which is which? Which part is a gene? Which part is a junk one? Okay. Next one is classify the junk DNA as intron, untranslated region, transposons, dead genes, and regulatory elements. So in this, what they're trying to say is junk means it is not waste. If it was waste, why will it be there in our body, right? These DNA, junk DNA, can be categorized into intron, untranslated region, transposons, dead genes, regulatory elements, etc. Also, what they would like to answer is how to divide a newly sequenced genome into genes, that is coding and non-coding genes. So all these things can be answered if we have a method to do genomic analysis. Now, the importance of genomic analysis can be understood from this slide, okay? What if you compare human and chimpanzee genes? I hope you can see there is a half chimpanzee and a half human, right? What if you compare the genome of these two? You will be surprised to know that the chimpanzee and human genomes vary by an average of 2% genomes, okay? So 98% of our genomes is same. So only 160 enzymes that we produce is different, okay? So then what happens? How do we look so different? Okay, what happened? So all those things, that what is the structural difference? What are those codes that are different in our DNA? Everything we'll be studying in the genomic analysis. So complete genome analysis of the two genomes, that is of chimpanzee and the human beings, would give us a strong insight into various mechanisms responsible for the differences. Now this is just this is a chart to give you an idea about different genomes and the number of genes present plus the genome size. Yeah. So here I'm giving you species, here genome size and number of genes. So you can see all these species over here, the genome size in mega basis and the number of genes present over here. Now what you will notice over here, I'll be taking it in the next slide, you're seeing Arabidopsis thaliana. Its genome size is 125 MB, okay? And the number of genes present is 25,500. Then you come to Homo sapiens, that is us human beings, okay? Human beings' genome size is 3,300, almost 250 times that of Arabidopsis thaliana. But the number of genes present is 27,000. Here it is 25,500, 27,000. So what I'm coming to tell you is Arabidopsis, it's a small plant, okay? Arabidopsis and humans have the same number of genes, okay? Though the Arabidopsis genome is around 250 times smaller than humans, how is that? So basically, I told you about the junk DNA, right? Human genome has a lot of junk DNA, specifically the transposons and the mobile genetic elements. So this increases the size of human genome, though the number of genes is only 27,000. So that you can see the size, and so you cannot say that, you know, there's a huge size means there will be comparatively more number of genes. You cannot say that. There might be a lot of junk DNA